I'm going to be presenting the phase 3 study of Taselisib plus fulvestrant versus fulvestrant and placebo in patients with estrogen receptor positive, PI3 kinase mutant, locally advanced or metastatic breast cancer. This is the primary analysis of the study that is known as the Sandpiper. Now, the background for this study, just briefly, as you are aware, the PI3 kinase signaling is deeply involved in breast cancer. It controls tumor growth, proliferation, and survival. And mutations are quite frequent of the PI3 kinase itself in ER-positive breast cancer. About 43% of tumors have that. There is a plethora of preclinical and clinical studies that support that this is a target for breast cancer. And the compound today is Taselisib. This is a beta-sparing PI3 kinase inhibitor that has high affinity and selectivity against the mutant isotypes that are present in breast cancer, which are PI3 kinase alpha. This is the first study, placebo-controlled, randomized, that was evaluating the efficacy and the safety of this inhibitor, Taselisib, when added to fulvestrant in patients with advanced disease. And importantly here, this is a biomarker-defined population. The primary endpoint was based on patients that had tumors with PI3 kinase alpha mutations. So these are the eligibility criteria. These are patients that had ER-positive disease, that had progressed to an AI, to an aromatase inhibitor. These patients could have received up to one line of chemotherapy for metastatic disease, and no prior fulvestrant, PI3 kinase inhibitors, or mTOR inhibitors were allowed. Patients' primary endpoint were patients that were PI3 kinase mutant, that's on the top, and patients were randomized 2-to-1 to receive Taselisib or fulvestrant versus placebo and fulvestrant. And then there was an exploratory cohort, and this was in patients that did not have tumors harboring PI3 kinase mutations. Primary endpoint was progression-free survival in patients based on investigators' assessment. And this is the primary endpoint. So the study is positive. The hazard ratio is 0.7. And the median PFS by investigator assessment goes from 5.4 months on the placebo arm to 7.4 months on the fulvestrant arm. Now, this is the primary endpoint. I'll present tomorrow the secondary endpoints, such as response rate, clinical benefit, endocrine response, and they're all consistent with the effects that we see here. The survival data is still immature. Now, what's interesting also in this study, and this is food for thought, the study was done, and this slide is not in your press materials, but I think it's an important slide that I'm happy to share with you. The study was global. It was done all over the world. And you will see that the data in Western Europe, USA, Canada, and Australia, and also in Asia, is quite different from what we saw in the rest of the world. So here in the Western Europe, USA, Canada, and Australia, it was 4.5 to 7.9 months, and the hazard ratio was 0.7. And it is little shocking, and I think we need to deep dive into this. These are countries that were mostly Eastern Europe and South America, in which we did not see any benefit at all. In terms of the safety summary, the addition of Taselisib led to a very clear increase in toxicity. You have an increase in the serious side effects. You have an increase in the grade 3 side effects. And the leading toxicity was mostly gastrointestinal, and this is probably due to the gamma effect of this inhibitor, with diarrhea being perhaps the most frequent one. Hyperglycemia is an on-target effect that was expected. So, in conclusion, the study met its primary endpoint. The median PFS went from 5.4 to 7.4 months with a hazard ratio of 0.7, and this is statistically significant. The secondary endpoints also were consistent with the improvement in the primary endpoint. Survival is not mature. The safety profile was as expected, but substantial at the level of the gastrointestinal toxicities. And then I'll say that if I had to give you a final conclusion, these results are positive, but I think we all would agree that they are modest. And this is a challenging combination from the point of view of tolerability, but I do think that this establishes proof of principle. This is a bona fide target for sure, but clearly in the case of this study, toxicity has to be weighted against benefit. Thank you.
Okay, thanks very much, Jose. So to offer some context and perspective on this study, I'm happy to ask Dr. Harold Burstein from the Dana-Farber Cancer Center for some comments. Hal?
Thanks, Rich. So this is a very appealing target. It's a mutation that is probably the most common in breast cancer when you do genomic sequencing, and it arises in other tumors as well. In fact, there are data at this meeting looking at basket trials that also have drugs that target PI3 kinase. And for over a decade, Dr. Baselga and colleagues like Carlos Arteaga and others have really worked incredibly hard to winnow out the mutations and try and find a targeted therapy to put together to show clinical benefit here. And I think what's interesting about this study is it shows that we finally have a lock and a key that we've begun to open the door. And the dilemma is that as soon as we opened the door a little bit, we ran into one of those latch locks that has that little chain, and it kept it from going further. So as Dr. Baselga said, I think it's a modest step forward, but it's an important step forward because it does suggest we can effectively target the pathway, and hopefully this gives us something we can build on so that we can use a target and a precision approach in this very common type of breast cancer subsets.
Thanks. I think you all should also recognize that I believe there are two drugs in this same general drug class, PI3 kinase inhibitors, that are already FDA approved for treating hematological malignancies. And this would be the first demonstration of a drug in this class having effectiveness in treating a solid tumor, albeit with the modest outcomes that we've discussed. Jose, I had one quick question for you. I'll take the moderator's prerogative to ask a quick question. So as you pointed out, the study used a biomarker selection strategy. Do you think that the biomarker selection actually identified the patients most likely to benefit from the treatment?
It did, although I did not show the data. There's also benefit in the non-biomarker patients. Now, it could well be that the assay that we use only look for specific mutations, and there are many others. So maybe those that benefit from the that are not selected, maybe because they had mutations that we did not pick up. So I would say the benefit appears to be greater in those that have mutations, but there is also some benefit in those that do not, and this is I think that need to be studied.
So the biomarker is imperfect.
It's imperfect.
Yes.
Yes.
Okay. Great. Thank you.